In this video, we're going to go over size exclusion, ion exchange, and affinity chromatography. Size exclusion chromatography, which is also called gel filtration, separates molecules based on their size. We start off with a solution that has a variety of molecules in it that have different sizes. We call this the mobile phase. We then pour that solution through a column that has a bunch of solid beads in there, and those solid beads are called the stationary phase. These beads are what actually separate the molecules, and what allows them to do that is the presence of a bunch of these pores throughout the beads. Molecules will move into those pores and get temporarily trapped, and the smaller the molecule is, the more likely it is to get trapped. Eventually, the molecule will make its way out. Larger molecules can't fit into the pores, so instead of getting trapped, they will move right past the beads through the column. After we pour our initial sample through the column, we continue to pour more of our mobile phase through the column to wash out any molecules that get trapped. And we do that until all of the molecules that we need have been washed out of the column. Here's an example graph that size exclusion chromatography might produce. On the x-axis, we have the elution volume, which is measuring how much volume of the solvent did we pour through the column. Since those smaller molecules get trapped in those beads, they require more of that solvent to get poured through the column in order to make their way through it. When you look at this graph, each peak represents different molecules that have made their way through the column. The more volume required to generate that peak indicates it's a smaller molecule. The absorbance on the y-axis tells you what quantity of molecules are in that peak. For example, if we look at this middle peak, the molecules that produce that are larger than the molecules that produce the peak on the right, but smaller than the molecules that produce the peak on the left. And since this is the tallest peak, this one has the highest quantity of molecules. In ion exchange chromatography, we are trying to separate the molecules in this solution based on their charge. There are two types of ion exchange chromatography: anion and cation. But we're going to work through anion exchange, and then you apply those exact same principles to cation. In anion exchange, all of the beads have a positive charge. What this does is allows all of the positively charged ions in that solution to pass right through the column and exit out, while the negatively charged ions move through but then get stuck to those positively charged beads. For example, let's say that this solution has proteins, and some of those proteins have glutamate amino acids. The glutamate side chains will be attracted to the positively charged beads and get stuck to them. Once we pour the full solution through the column, we've successfully isolated all of the negatively charged molecules. But now we have a problem because they're stuck to these beads. The way that we get them unstuck is to pour a new solution through the column, and this solution has some salt in it, like sodium chloride. Those chloride ions, or whichever anion is a part of the particular salt used, will compete with whatever anions are already stuck to the beads, and they're going to try to kick them off of the beads. One cool thing about ion exchange chromatography is that not only can you separate these things based on their charge, but you can also separate molecules with the same charge based on the magnitude of charge that they have. Let's say that protein 1 only has the single negatively charged glutamate side chain, but we have a second protein that has two negative charges on it. That second protein will have a stronger bond to those positively charged ions. To separate these two proteins, what we do is we start with a very low concentration of our sodium chloride solution. We pour that through the column, and that's going to wash out the molecules with a negative charge of the smallest magnitude. For example, let's say this one washes out molecules with a -1 charge. And then we gradually increase that salt concentration. And just for example, we'll say that the 0.05 concentration washes out those with a -2 charge, and the 0.1 washes out those with a -3 charge. And these molarities, by the way, are just completely arbitrarily chosen. It's just the concept that you need to have a really good understanding of. And the reason we call this anion exchange is because originally we have an anion that's stuck to the beads, and then we exchange that with a new anion. For cation exchange, everything is the exact same except you just flip the charges in all the situations. The graphs for ion exchange chromatography can have a variety of different looks, but here's one example. At the x-axis, we have time, which is just showing us how long it takes for these molecules to come out of the column. And then we have this red line, which shows us that as time goes on, the salt concentration that we're pouring through goes up. Each peak represents molecules that have left the column, so the earliest peaks represent molecules with the lowest magnitude of charge, and the later peaks show the molecules with the highest magnitude of charge. Once again, the y-axis, which is fluorescence, shows us how many molecules are in each one of those peaks. Lastly, we have affinity chromatography. In affinity chromatography, The beads are specialized to trap one type of molecule. Sometimes this is done by putting enzymes on the beads, and they're going to trap the substrate that fits into that enzyme. We might put antibodies on the beads, and as you know, antibodies are very specific for certain molecules, so they will only catch those molecules out of solution. And the other type of chromatography, which comes up a lot on the MCAT, is called nickel purification. This is the one that we are going to talk about for our example. This is a technique used to separate a specific protein from a solution. Way before we're actually at the chromatography process, we engineer a DNA sequence to add a bunch of histidine residues onto the protein of interest. And that's why you'll also see this technique called his-tagged purification. The proteins that we engineered to have those histidine residues are in this solution. And then the beads in this column have this nickel 2+ ion on them. What I've drawn here is an oversimplification of what's on the beads, but you really don't need to know any more details than this. As the proteins of interest pass through, these histidine residues are going to use that lone pair on the nitrogen to form a coordinate covalent bond with those 2+ nickel ions. And this is actually happening with 2 histidine residues on the protein, but again, that level of detail is not something you need to know. What this ensures is that the only thing trapped in the column is the protein with those histidine residues. All of this shows that affinity chromatography is great for isolating exactly what we want. But the downside is that since it's so specific, these molecules are now attached to the beads with really high affinity, and it's hard to get them off of the beads. There's a variety of ways to get these things off, and a common way that you want to look for on the test is to pour something through the column that's going to compete with whatever's bound to the beads. For example, if we have enzymes on our beads that have trapped their substrate, we might have a molecule that looks like the substrate to then kick those off of the beads. For nickel chromatography, you can also use proteases, which then cleave that histidine tag and let the rest of the protein leave.